Thank you very much for inviting me. And for me, it's a pleasure to give this talk at the TRI Academy. And I would like to start showing you some of the images that we have been able to obtain. And these are neuron-like cells from a patient with tinnitus and with a rare variation in the gene ANG2. So in this panel that you can see in black and white, on the left, we have control cells. These are neurons, the neuron-like cells, we have to say, that have been derived from an asymptomatic individual. And on the right panel, we can see different structure, and you can see how the main feature that we can see in this phenotype is a high number of connections that these neurons are developing when we compare with what is happening with the control cell line in vitro. And this is for me quite exciting because it's the first time that we are starting to see really changes at the cellular level. So I would like to organize the presentation. And so the first part is going to be about clinical research. What are the evidence that we have been able to collect to demonstrate that tinnitus has heritability? And I will make an overview about the different strategies. And the second part will be about the genetic data that we have been able to generate and compare to the different type of approach that has been followed. No, okay. So the first thing is that we need to consider that tinnitus can be a symptom, which is something which is very common, and it's something that is associated to many conditions. So probably that's one of the major difficulty to investigate tinnitus is that we really need to define what are we dealing with because not all the individuals has the same etiology and the same origin. And one of the strategies that we are following, and we need to distinguish tinnitus as a symptom from tinnitus as a disorder. Tinnitus as a disorder is a multidimensional condition that is usually associated with either emotional stress or cognitive dysfunction, autonomic arousal, and can be also associated with behavioral changes. And all of this is leading to a functional disability that at the end is probably the main issue for the patient and for the individual. So the patients perceive tinnitus as suffer from this, and the individuals really needs to look for help and support. So tinnitus is a perception of a phantom symptom of an auditory noise that is usually perceived in the head, but could be also perceived in one of the ears. And it's affecting 15% of the population. However, tinnitus disorder, fortunately, is a more rare condition and is usually only involving 1 to 2% of the population. The thing is that tinnitus overlap with multiple disorders, and this is also something that occurs with tinnitus disorder, and we have a very complex heterogeneous phenotype. So the different evidence that we have to support tinnitus irritability come from epidemiological studies. Epidemiological studies are very relevant to demonstrate for any disease that you have a genetic contribution. Several types of studies has been performed, mostly in Sweden, with twin studies. So Swedish twin cohort has been compared. It has been demonstrated, as you can see in the diagram, that monozygotic, that means identical individuals for their genome, have a higher concordance compared to dizygotic twins. And this concordance is particularly higher when we try to get a better definition of a tinnitus, and this is when patients have bilateral tinnitus. Unilateral tinnitus has a low irritability. And this is something also that if we select individual with a young age of onset, the irritability increases. The same finding has been shown in adoptive studies. So the degree of concordance that the adoptive has is higher with the biological parent than the adoptive parents in this type of familial studies. There are several types of genome-wide association studies that are based on self-reported tinnitus that have been associated also, association with common variants, and also familial aggregation study has demonstrated that tinnitus, it has a strong familial aggregation. This familial aggregation is shown here. Is higher for severe tinnitus and it was higher in women. Just to give you a rough number, so the estimation of the familial aggregation is 10 times higher for severe tinnitus in women and 5 times higher compared to the general population for severe tinnitus in men. And this is particularly relevant if you start to see what is for bilateral or unilateral tinnitus, if you include any type of tinnitus. So defining the phenotype increase the chance to have a genetic contribution. So that means that we have really to define the phenotype for genetic study. That's the first conclusion that we have. And this has been, in fact, the major limitation of many genetic studies performed in tinnitus. Tinnitus was self-reported. This is what happens when we collect data from biobank. So biobank data are not designed for study any particular disorder, or in any case, they are designed to study common disorder, but not when you try to identify individuals with rare disorder or individuals with complex phenotype that have overlapping phenotype with other conditions. Self-reported symptoms are not enough specific to define the phenotype. And that explains the lack of replication of most of the GWAS that we has been performed. And probably the study that was followed like 20 years ago using candidate genes cannot be selected because usually there's problem with the selection of the individuals stratification of the cohorts. So precise phenotyping is the best strategy, and there are only a few elements that we have to consider into this. So the first is the age of onset. As occur for most of the genetic conditions, the early the onset of the disease, the highest the chance that we have a genetic contribution. The ethnic background is essential in all the studies because we know that the conditions usually change according to the geographic location. And also hearing loss. We know that there is a strong association between hearing loss and tinnitus and hearing loss, hyperacusis, and tinnitus. So these three conditions are clearly interconnected. And this is something that we have to get specific data to try to establish what are the contributions for each of these dimensions. And for that, the best way to proceed has been using a psychometric assessment of patients. And the preferred tool has been tinnitus handicap inventory, tinnitus functional index. So these are tools that can be used to define how is the distribution of the phenotype across multiple individuals. And the strategy that we have followed to understand how the phenotype can change across the different individuals is the selection of the extreme phenotype. What is the extreme phenotype? We have to assume that the symptom, despite they have a multi-dimension involvement, are following a normal distribution, and they can also follow Poisson distribution. So we will have in one of the extreme individuals with a benign phenotype. So these are individuals with mild symptoms. In the other extreme, we will have individuals with severe phenotype. The majority of the individuals are not going to be in this both extreme. The majority of the individual will have an intermediate phenotype. And including all these individuals can generate a lot of noise in the genetic data. So we have to be able to measure the phenotype and selecting individuals that are in one of the extreme of the phenotype in this case has been a successful strategy to identify genes. And as I was mentioning, we have been selecting these individuals according to the tinnitus handicap inventory, which is a very simple questionnaire with 25 questions and very easy to collect, and particularly in clinical setting with a great overload of work, is something that we have to consider, something that can be feasible and implemented in the clinical practice. So this questionnaire goes from 0 to 100, and this is a way to standardize the score across the distribution of the phenotype. And this is just to show an example, and as you can see here, this distribution of THI score really does not follow a normal distribution. We had a significant number of patients with Meniere disease that either have no tinnitus or either have a tinnitus that has no relevance for them. However, since we don't have a normal distribution, we can define quartiles uh, to determine what are the number of individuals that we have in each part of the distribution. And here, the ones that are in dark green are the individuals that had a THI score over, as you can see here, 68, which is really a very severe score for tinnitus. So I would like to try to explain what is the relationship between the genome structure, the variation, the contribution to the genetic variation, and the prevalence of any disease. In fact, I will go to the particular case of tinnitus. So we know that for most common disease, the contribution of the genetic variation is usually associated with common or with rare disease, but usually it's common variants. However, for rare disease, common variant has very little relevance. The most common conditions are that rare variants are associated with rare disease. And this is the example that we have. Tinnitus as a symptom is a very common condition, and it's likely that it's going to be associated with common variation. We have a common disease associated with common variation. However, tinnitus as a disorder is considered a rare disease and is going to be associated with rare variation. And the most important thing here is that the effect size of rare variation on the phenotype, on the individual, is very high. However, the effect size of common variation in the phenotype is very small. So if we want to do any intervention in the future using this genetic structure, the target should be the genes that are the carriers of the rare variation, because a change in a single gene is going to be a change in the phenotype, a big change in the phenotype. I mean. So for this, we have been followed different strategy across many years. So the DNA sequence can be read, can be defined using a genome-wide association study that is a genotyping approach. So we are not reading the complete sequence. We are only reading some of the base of the sequence, some of the letter of the text, and we are imputing the rest of the variant. However, using this approach, we can study coding region and non-coding regions, but we are not able to have a high resolution with the rare variation. And that's one of the limitations of genome-wide association study. The current strategy is to use exome sequencing or genome sequencing. Exome sequences read the full coding regions. Genome sequencing read the full genomes, the coding and the non-coding region. So how will be the sequence if we try to read the sequence with a genotyping approach? That is the one that has been followed in genome-wide association study. So as you can see here, we can identify some of the letters in the sequence, but we can nearly understand. We have to make an imputation and try to make an estimation what could be this all X or this all Y according to the next base that we have been able to read. The exome sequencing is an approach that we are able to read the exome. The exome are the coding regions, and we can understand or we can read the whole coding regions, and we know that the coding regions accumulate the majority of the rare variation. And that was really a very successful strategy for rare disease. So we can fully understand some of the words here, but we cannot really know what is the contribution of the non-coding region. And could be extremely important in certain cases to explain particular differences between individuals where we have different expressivity. So maybe in a single family, we can have individuals with more severe or with more mild phenotype carrying the same rare mutation, that we have to look into other common variants to explain the difference in that case. However, that's why the preferred strategy is to do right now genome sequencing, because we can understand what is the contribution of the coding and what is the contribution of the non-coding regions. So just to understand and to go again to the same scheme that I was trying to explain, GWAS has been studied and could be useful to identify the genetic contribution for tinnitus as a symptom, because the resolution of the GWAS is restricted to common variation. However, if we want to define what are the rare variants that contribute to the phenotype that I have mentioned, they have a large effect size on tinnitus disorder. We are using a strategy which is the burden test. That means the accumulation of rare variation in certain genes. That is the way to target the gene, because this is not a Mendelian disorder. Has to be clear. Mendelian disorder are really more simple, we can say. Here we have different rare variants across different individuals, but we are trying to target what are the genes that accumulate this rare variation. And this is just a summary table of the different genome-wide association study that has been published for tinnitus. And we here consider tinnitus as a symptom. And as you can see, I have tried to highlight in yellow 3 variants. And this is the the 3 variants that have been replicated. And as you can see, many of the variants that has been replicated, and some of these are from the UK Biobank data, they had not been replicated in a second study using UK Biobank data again. So different groups have been trying to use the same data, but they are not replicating the result. So probably the reason for this is that this is not the best strategy. And as you can see, the second limitation is that the allelic frequency, if you look at the right of the last column, you will see that the minor allelic frequency of this variant is very high for 2 of them. So that means that this is very common in the general population. Only one of the variant is really very rare, and this variant could be relevant, but it's the only one that has been found using this strategy. So the different things I'm going to present right now are part of my former PhD student, and this was the work of Sana Amanat that was working in this project in the EXIT network in Europe. She was working during one of the visits. This picture was during one of the visits that we have at the Institute of Mario Negri. And I think that this is a paper that we published a few years ago. And the design of this study was to select very few individuals that they have a very severe tinnitus with a very high score. So the tinnitus extreme phenotype was defined in 30 individuals with Meniere disease and severe tinnitus. We also selected a second subgroup of patients with 29 individuals, what we tried to use to replicate before. And this is what we call the almost extreme phenotype. So this is like the 3rd quartile, and the extreme phenotype is the 4th quartile. And this we compare this also with patients with Meniere disease without tinnitus, that we use this as an internal control, because this is the way to identify what are the particular genes that have the burden of rare variation that contribute only to tinnitus and not to the whole phenotype. And we use different set of controls. I'm not going into that. This is a public reference data set that we obtained from NOMAD and also from the Spanish population. And also we use 2 validations control set group, one from Swedish individual with tinnitus from the stop cohort, that we selected 97 individuals, and we use more than 700 individuals from Germany with genetic generalized epilepsy as external control, because this is new, has no tinnitus at all. And to make this story short, I'm just highlighting the genes that we were able to replicate. So this table, which is very busy, I know, this is all the genes that we found an association in the discovery cohort that was performed in Spain. But only these three genes that I'm marking here, AKP9, ANG2, and TSC2, were the three genes that were replicated in the Swedish cohort. And this is what I'm trying to put it here. So these were the three genes that this is, because the Swedish cohort are patients with severe tinnitus, but they don't have any Meniere disease. So this is how we started to define that this particular genes has few rare variants, but they have a strong, a big effect size on the phenotype. This is the distribution of the variants that you can see here in the Meniere disease control in the Swedish population in yellow and in red in the three genes. And this is a summary table so you can have an idea what is the effect size on the phenotype. So these are the variants, these are the genes, and we have selected the variant that have the higher effect size for all of these that are associated with the tinnitus disorder. And you can see that they increase in a single individual more than 10 times or more than 15 times if you are carrier of this variant. However, when we look at the variant that has been associated with tinnitus as a symptom, the effect size is very small, usually below 1.5. So that makes a big change in terms that the frequency is very high of this variant. So this is shared for many individuals in the population. Many of them has not been replicated, which is really a major concern, and the effect on the phenotype is very small. And try to understand what could be the effect of this rare variation. We did a gene ontology analysis, and we found that the biological processes that were enriched with higher significance were the synapses and the cytoskeletal binding protein. So it was very clear that we're going to expect the finding genes in the synapses, but we were a little bit surprised because we were expecting receptor, we were expecting adapter protein, but no cytoskeleton proteins. And that's probably explained that some morphological changes are expected in the carrier of these rare variations. So this is the conclusion of the study that was the PhD project of Sana. It was clear that we get replication in three genes in the Spanish and Swedish cohort, and they was not replicated in patients with generalized epilepsy, as I have mentioned. And what is the function? And I'm just going to take the model of ANG2. So we think for several experimental data that has been already published that ANG2 could be related to the process of elongation of the axon. So ANG2 is related to the growth of the intra-axonal boundary. So it determines the length of the axon. So the lack of auditory input is possible related to the increase of the length of the axon initial segment, which is a specific part of the axon, and is the region that determines the growth of the axon, of the distal axon. And also, there are some functional cellular study that has been performed in the knockout mice for ANG2. And this knockout mice is a model for autistic spectrum disorder. And it has been shown that the number of projections that the neurons has in the knockout mice are different. There's a increase in the axonal branching and the generation of ectopic neural connectivity. So that means that the loss of function of this gene is probably related to increase in connectivity and increase in the number of excitatory synapses in this mouse model, because this is mouse model. So the general hypothesis that we are trying to generate with this is that this enhance of connectivity, which is something that has been already proposed by functional imaging studies is probably the underlying molecular and cellular process involved in tinnitus persistence. And I'm going to show you some of the data that we have done, and this is before. So the next step for us has been to select patients with these mutations in ANG2, and we have been able to generate a neuron-like cellular model. And this is like a standard process, and we have been able to compare how is the growth across different during the process of differentiation of these cells and compare this with the cell lines. So the first two rows in this slides are the carriers of the one of the missense mutation that we have in ANG2, and the two last row are the control cell line. And as you can see in the panel B, you can see that there is an increase in the density of the neurons in the carriers of this mutation in ANG2 that is not observed when we compare this in the individuals that have they don't have the mutation. So that's also interesting because the distribution is quite different. And this is again another imaging that we can see the organization of this cell is quite different when we compare the wild type, when we compare the control cell line with the carriers of the mutation. As you can see, the organization is pretty different in this of them. So this is just an example. We cannot probably go further with this model, but it's like the biological basis to demonstrate that there is an increase in the connectivity, that something that was previously shown in the knockout mice, we can see also in cells that are derived from patients. And the next thing that we have been trying to do is try to define what are the clinical phenotype of this individual. Is something that we can identify from the clinical point of view. Well, this is something more complex to say, but one of the things that we have observed is that most of these individuals has a high frequency hearing loss, and I'm meaning something that is over 8 kilohertz. And also, most of these individuals have a severe phenotype, and also we are obtaining a score for cognition level. But the number of individuals that we have here is really very small because, as I has mentioned, we cannot jump into conclusion with 12 individuals because the number of individuals that are going to be carriers of a rare variation in ANG2 is also very small. That's the problem. But for this particular individual, this variant may have a large effect. And what are the common symptoms? Hyperacusis, persistent noise-type tinnitus, high-frequency hearing loss. And also, when we analyze the middle latency response, we observe an increase in the PA wave amplitude, which is something that was not expected. Some of the patients has also anxiety, depression, and cognitive symptoms, but that was not a consistent symptom. We don't think that we can really relate that to the mutation at all.
Sorry to interrupt you, Antonio, because we need to slowly move on because of the time.
Okay, so I will skip some of these and maybe I will just go to the conclusion if you want. So this is the genome studies. This is part of the study that we have continued analyzing data. And we have included not only single nucleotide variant, we are also been trying to integrate a structural variant. This is really very, very large to detail, but a structural variant are also contributing to the phenotype, to the extreme phenotype. And I will skip all of this to saying that also we have been able to identify variants that are protective. So I'm trying to say not everything is bad news from the genetic perspective. Also, we have some variants that can contribute to develop some resistance to tinnitus. And I will not go in detail for that because of the short time, but this is also something that could be related to some changes in the structure of the organ of body, particularly some structural changes in the territorial membrane, and some mechanism to protect herself to develop some resistance to tinnitus. This is something of an area of interest. So the take-home message, as I will finish very soon, as you asked me, is that we have now evidence to support that there is a cellular and molecular basis for severe tinnitus, and the most strong evidence that we have is for the ANG2 gene. But we know that this is just the beginning of the understanding of this complex genetic structure, because we have also been able to obtain evidence that a structural variant would have also explained part of this severe phenotype and even part of this unexplained common phenotype that have any tinnitus symptom. So this is something under review, something that we are still analyzing, but we have some preliminary data on that. And also variation related to other few genes may have a protective effect to develop severe tinnitus. I just want to finish. Thank you to all the people that has been involved. That not only Sana, also Alvaro and Alba has made a strong contribution. And now Alberto is continue analyzing the phenotype of this patient, and Patricia, of course, in Granada. And this is my new team in Sydney. And of course, I need to thank the knowledge and the Unity support and all the people in the Unity network in the different projects, and also particularly Christopher with all the heritability studies that were conducted in Sweden. Thank you so much, and I try to finish as soon as possible.